Enzymes inhibitors. Inhibitors, you can say that these are the enzymes which do not work properly. Why they do not work properly, or they are actually decreased to perform their activity. So this kind of, when the enzymes have decreased their activity, it is called enzyme inhibition. Enzyme inhibition has three types or three ways you can say the enzyme can inhibit it to perform or catalyze its function. One is competitive inhibition, second non-competitive inhibition, and third one is uncompetitive inhibition. So what is inhibitors? Inhibitors, you can say the substances would decrease the activity of enzymes, so enzymes cannot perform their functions. Or in other words, you can say that enzyme inhibitors are molecules that are bind with enzymes. So when these molecules are bind with enzymes, so enzymes cannot perform their activity or reduce their activity, you can say. Both things are possible with it. So as per the types, you can see I have already seen the competitive inhibitors, non-competitive and uncompetitive. All three kinds are actually in enzyme inhibitors. So first one is competitive inhibitors. What happens in the competitive inhibitors? The competitive inhibitors, the inhibitors directly, you can say, bind directly to the active site. Whenever the active site is directly bind, so catalyzation cannot be took place. What happens in this type of inhibitions? Actually molecules which are exactly similar like the substrate, but they are not the original one. They are just similar, just the same one, like a original substrate. What they do, they actually bind the active sites. So original substrate cannot be fixed into or cannot take a active site, so the actions cannot be took place. So competitive inhibitors, here this type, in which the molecule which are exactly similar like a substrate, which are not the original one, they actually bind the active site. So original cannot be fixed and catalyze the reactions. So as inhibitors block the enzymes, so responsible for this kind of mostly we have seen in a penicillin. Penicillin is antibiotic where we have seen that in the human cells, whenever they are not required one, so binding is took place in as per the competitive inhibitors. Examples, you can say methotrexate to for cancer is a common example for it. Non-competitive enzyme. In a non-competitive enzyme, what happens in this one? In non-competitive enzyme, the inhibitors bind to the different site, not the same one, but they are just a similar one, which we call the allosteric site. They actually bind the different site and change its shape. So enzyme shape is changed or substrate is changed, shape is changed, so it cannot be fixed. So you can say simply in this, non-competitive inhibitor is a type of inhibitors when molecule actually bind with enzyme. When the molecule bind with enzymes, so instead of active site, here active site is not bind, here the enzyme is bind. When the enzyme is bind, so molecules, such molecules are called non-competitive inhibitors. Why? Because these binding sites are always from active site, which we say that allotropic site, because these are the different shapes giving to it is because of binding the molecule with enzyme. Interaction causes, so enzyme cannot actually catalyze the reactions because of its shape change, and when the shape is changed, the reading of the active site is not possible for it. Next one is uncompetitive inhibitor. Actually what happens in uncompetitive inhibitors, the inhibitor bind only to the enzyme substrate, complex and stop it for reaction. Here it is also known as anti-competitive inhibitor. Uncompetitive inhibitor is also called anti-competitive inhibitor. It takes place as already I told you, enzyme inhibitor actually bind the enzymes for complex format, or you can say it can bind enzyme substrate complex, enzyme and substrate. So they are bind enzyme and substance complex because both are bind. This type of inhibitions you typically occurs in reaction with two or more substrate, where the two or more substance are produced or available. So this kind of inhibitions took place. Okay, what's the significance, or you can say importance of the enzyme inhibition? Why they are required one? What is the need for it? The importance of enzyme inhibition in a medical field, so much. Medicinal drugs like that, you already I have told you about that penicillin, angio, strepsin are treating the illness. How they are actually, they are becoming inhibitors. They actually are inhibiting the enzyme to perform their function in a bacterial one. So major mechanism, you can say, a biological system especially occurs in small molecules. Why small molecules? You have already known that the identified substrate and its condition or critical for a catalysis. These enzymes are various levels for and diseases, and stop the growth when by the inhibitors. Feedback inhibitors is the activity almost where the inhibition is took place by permanently disable the function. This type of biological mechanism or activity, you can say, see the growth of its own product. So it is called feedback inhibition. Reversible is a non-competitive, and it's mostly see whenever the product is in high quantity. Whenever the product in high quantity, they actually bind completely enzyme size. So this kind of actually done due to the product is consumed and the inhibition is reduced. Production, when the production is consumed, fully consumed, the inhibition is also reduced and form more products. When this kind of things is seen, the feedback inhibition will call it. Whenever you will see the product path is end, product path is end, now no more way or is seen for the product to be formed. The last one will change or will combine with another site, so they will not give away again for a more product, or end of the product is called feedback. It is a binding shutdown path. Mostly we called it a shutdown path. That's why it is feedback inhibition. You mean permanently disable it. Thanks for watching.